Thank you so much for having me, and I would also very much to thank Danny and Danielle for the invitation to be here. It's really a both privilege but also humbling to be with all of you and hopefully be helping as part of the overall ecosystem to bring new treatments to you all. I'm an internal medicine physician, scientist. I have, in addition to taking care of patients, very early on got involved in developing therapeutics. I've actually worked across small molecules, gene therapies, and today I'm going to be talking to you about oligonucleotides. And as part of that background, I've actually had the privilege of bringing 3 of those products through the Food and Drug Administration. You may be wondering, CAMP4, what's the background around the name? So CAMP4, for some of you mountaineers, may know that it is the final prior to the summit camp before getting to the top of a mountain, particularly of Everest, and we use this as a motivator for our team in terms of bringing new treatments and all the hard work that you've all heard about that goes into developing new treatments. This next slide is required by my legal department to share with you all. And my disclosures is that I'm an employee of CAMP4. So CAMP4 is actually an 8-year-old company. We were spun out of the lab of Rick Young at the Whitehead Institute, that's part of the Massachusetts Institute of Technology in Boston. And Rick in particular is an expert alongside a collaborator of his named Len Zahn at Boston Children's around really trying to understand what controls transcription, what controls making ultimately the RNA from DNA. And so the company was set up and has actually been at this for now 8 years. And so what I'm going to talk to you about is an antisense oligonucleotide that we are actually developing for SYNGAP1. We actually have a developmental candidate that we've declared, and I'm going to show you some data, but we'll give you a little bit more background. Antisense oligonucleotides have been used before, and I'll show you some of them that have actually been improved, and they can be used in different ways to actually control expression. Many of these have actually been focused on degrading or reducing protein expression more in like gain-of-function type diseases. We, however, have developed an ability to actually map what are the control elements of different genes and then actually use that to also upregulate. And I'll talk a little bit more about that. And so we're developing this ASO for SYNGAP to actually increase the amount of SYNGAP protein which is absent. Before I mention some of the ASOs that are approved, one thing that I'll come next to is you can see, and this has been mentioned quite well in some of the prior talks, around you have DNA and then pre-mRNA and then mRNA and then protein. Some ASOs, particularly in genetic epilepsies, are focused on taking pre-mRNA and actually increasing through splice modulation the amount of then the final mRNA before it goes to protein. Where we're focused is actually at the development of taking the transcription of the DNA to the pre-mRNA. So it's a slightly different area of functioning. But I think what's also really important is that we now have a lot of experience around antisense oligonucleotides across different diseases and also across different delivery mechanisms. We actually, at CAMP4, are running another trial that I'll mention briefly in Australia that's focused on a metabolic disease in the liver. And so we actually administer that subcutaneously. And so the benefit of having these other ASOs that have been improved is we can understand the chemistry and the safety profile and use that same chemistry. And that's really important to us, is we're not actually creating new chemistries. We want to use existing chemistries that we know are safe and that we understand how they can be used safely in trials. I won't spend much time here because this was shared previously, but the intrathecal route is one of the main routes that we can use to get this into the brain. This has become quite common. It is obviously not without risks, like any procedure has risks, but it is something that neurologists do all the time. And one of the benefits is it helps us make sure that we get the appropriate amount of the ASO to the relevant areas of the brain. And that's really important as we think about different diseases, because different regions of the brain may play more important roles in particular conditions. And so we want to be able to do that. The problem sometimes with administering it more peripherally, like either subcutaneously or in the IV is then the ASO goes to all the tissues of the body, and now you have to be concerned with what are some of those off-target effects. So SYNGAP is a haploinsufficient disease. In this situation, you have one good and then potentially one bad or mutated copy. And the bad copy may either not be expressed or may be expressed but then degraded very quickly. And so you end up with typically around 50% or so less protein. And this is important for the disease. What we at CAMP4, and I mentioned our founder, Rick Young, identified is that there are many components that are important for regulating the level of expression. And in particular, there are these kind of what previously were called non-coding RNAs. We have coined the term regulatory RNAs that control this. Some of them will activate or increase the amount of RNA that's produced. Some of them will decrease them, repressors. And what we found is that if these repressors in particular have a binding site that ASOs can bind to. So we create an ASO that's specific to that reg RNA. We bind to it. It actually changes the conformation of the repressor and causes it to come off the gene. And that is akin to taking your foot off the brake and the car would move forward, or in this case, the gene would increase expression. And so this can actually allow us to get to near normal SYNGAP levels, and we actually have data on this. Just maybe to illustrate this a little bit more, because there are different approaches, and I think we're going to see, and this is a very positive development, more ASOs being developed, but they may be being developed to target different parts of the pathway. Is you could think about an analogy of two dams, both at the top of a river. We're trying to fill the reservoir, and one of the dams is closed. So this is akin to one of the alleles not being mutated and not producing. So how do we fix this? So our approach is that we are going to have reg RNA ASO workers who can actually increase the size of the dam openings and allow more water to flow through. We're not able to increase the other one because it's mutated and it's closed. Another approach might be to, while you couldn't, for example, increase the size of the openings, you could remove some of the rocks along the way or change or alter the path. This might be akin to more splice modulation approaches. All right, so now I'm going to show you some data. So just maybe to cover with a little bit more depth the approach. So this is the chromatin loop during transcription. And as part of this chromatin loop, you can see the loop comes together and you have an active enhancer and promoter. And at that site is a complex that's responsible for transcription. And actually, this is very finely regulated. And so that's where the regulatory RNAs can function. And so when you look over the top of me, you can see, for example, in this case, this is the example of a repressor that might be on a particular active enhancer. And so we can bind to that, remove that, and increase gene expression. So how do we make these ASOs? We actually have developed a platform where we take many different cell types. Oftentimes, these are iPSC cells. And then we actually map all the regulatory RNA elements that are part of that cell type that are associated with particular genes of interest. And in fact, many of these genes have multiple reg RNAs. So we focus on identifying which are those that actually have a one-to-one relationship with the gene. We don't want to target a reg RNA that might touch many genes. We want to focus on that one single gene that reduces the likelihood that we could have off-target effects. We then actually use a variety of machine learning tools, initially in silico before we go in vitro, and we actually make multiple ASOs. We screen them electronically, and then ultimately we make them and we actually screen them in cell lines. And so we've been doing this for a while. As I mentioned, we have a clinical trial that's ongoing right now here in Australia for urea cycle disorders. This is a disorder that occurs both in children and adults where they have a poor ability to metabolize ammonia into urea. But our second program is SYNGAP, and I'm going to show you some data there. So on the left, you'll see here, we identified that there were in fact multiple SYNGAP1 enhancers, and we also identified multiple types of reg RNA that were responsible there. We identified a particular one that seemed to be most important for actually controlling overall expression. And so then we developed ASOs to target that. And what you can see in the middle inset here is that the ASO, when it bound to the reg RNA, increased transcription. And the way we measured this is by actually looking at increase in RNA polymerase II, which is a key machinery element for doing that. When we compared that, though, to housekeeping control genes, we didn't see that same element. And then to the far right, what you'll actually see here is now we're actually wanting to look at mRNA that's produced. And so we have a family or familial control. We then have a SYNGAP patient, iPSC. We, for the control, used a non-targeting control, so it's basically like a scrambled ASO that doesn't actually target. And then you can see here 2 different ASOs that we created, and you can see both of them actually bring the level back to wild type, and that's of mRNA. But what we really care about is protein, so what about that? Over to the left, what you can see is that using this this was in a mouse model where we replaced the SYNGAP mouse gene with a humanized mouse gene, and then we gave this ASO and we looked across different brain regions that are important here, and we actually saw a dose response, which gives us increased confidence then that ASO is actually producing the protein. And then to the inset to the right, this shows that there was also dose responsiveness in a SYNGAP mRNA with a mouse surrogate ASO. So this is a little bit different. Rather than using the humanized gene, what we actually did is created a special ASO that targeted this unique sequence for the mouse. The mouse sequence and the human sequence are a little bit different. So we actually create these mouse-specific ASOs as well. And so we're working very hard to actually bring this ASO to patients in a clinical trial as soon as possible. And it's going to be really important that we all work together as an ecosystem to be able to help get the trial up and running. One of the both opportunities but also challenges in SYNGAP is because there haven't been other studies of disease-modifying or genetic therapies. We need to work together to figure out what are the appropriate endpoints, how do we measure them, over what period of time. And so I just am really grateful for all the work that you're doing here for this conference to be able to allow us to do that. And then lastly, I'll just say we are very excited. We're going to actually be presenting some primate data for SYNGAP, and that's going to be at an upcoming conference in 2 weeks. It's the American Society of Gene and Cell Therapy. And after this, we'll be working to also disseminate that data as we start to move towards being able to go into clinical trials. So with that, I'm really grateful for the opportunity to be here. I'm grateful for the invitation, and I'm looking forward to getting to know you all better and working together with you.